The next topic is chromosome organization and molecular structure. Introduction. Now, what are the chromosomes? Chromosomes are the structure that contain genetic material, DNA or RNA. In eukaryotes and in most of the prokaryotes, DNA is genetic material. In viruses, few viruses they have DNA, few viruses they have RNA as their genetic material. So the chromosomes, they are complexes of DNA and proteins. The genome comprises all the genetic material that organism possess. In bacteria, it is a single circular chromosome, single circular double-stranded chromosome. In eukaryotes, it is a set of nuclear chromosomes. Eukaryotes also have mitochondrial genomes. Mostly animals they have mitochondrial genome. Plants also have chloroplast genome along with the mitochondrial genome. Remember, plants में दोनों भी होगा, chloroplast genome and mitochondrial genome. The main function of the genetic material is to store information and pass it on to the next generations. The DNA molecule does that through its base sequences. DNA sequences are necessary for synthesis of RNA and cellular proteins, that is for transcription and translation. Transcription is used for synthesis of RNA and translation is synthesis of protein using RNA molecules, mRNA as template. Then proper segregation of chromosomes during cell division, replication of chromosomes and compaction. So the DNA sequence is necessary for synthesis of RNA and proteins, segregation of chromosome during the cell division, mitosis or meiosis, replication of the chromosome, compaction of the chromosome so that they fit within the living cell. Viral genomes. Now the viruses are small infectious particles containing nucleic acids surrounded by capsid of proteins. For replication, viruses rely on their host. They cannot replicate themselves. They require host cell machinery. That is why viruses are known as intracellular parasites. Most viruses exhibit a limited host range. That is, they infect only to their specific host. They typically infect specific type of host or one host group. This is general structure of virus. Non-enveloped viruses which have capsid, protein capsid and nucleic acid present inside the capsid. This one is enveloped virus which have lipid layer around this nucleocapsid. Okay. Membrane. These spikes are of proteins, different proteins in different viruses, which helps in attachment of virus to the specific cell. Inside this membrane is capsid and inside the capsid is core which consists nucleic acids. This lipid bilayer is picked up when viruses leaves the host cell or when rupture the host cell and get released from the host cell. They pick up the host cell membrane which helps viruses to fuse easily with their host cell. Bacteriophages may also contain sheath, base plate and tail fibers. Viruses, they have genetic material also termed as viral chromosomes. The genome can be either DNA or RNA, either single-stranded DNA or double-stranded DNA, either circular or linear genome. Viral genomes vary in size from few thousands to more than 100,000 nucleotides. These are examples of different viruses and their host. So this lambda-phage, its host is E. coli, that is lambda-phages infect E. coli. Influenza viruses infect mammals. These are their genetic material. See, few of them have single-stranded DNA, few have double-stranded DNA, few have single-stranded RNA. Likewise, these are the sizes of different viral genomes and number of genes present on it. Then the bacterial chromosomes. The bacterial chromosome is found in a region called nuclei. There is no specific nucleus structure present inside the bacterial cells as like eukaryotes. So the region where this bacterial genome present is known as nucleoid. The nucleoid is not a membrane bound. So the DNA is in direct contact with the cytoplasm, or DNA is suspended into the cytoplasm. Bacteria may have 1 to 4 identical copies of the same chromosome. The number depends on the species and growth conditions. Bacterial chromosomal DNA is usually circular double-stranded DNA, that is few million nucleotides long. E. coli, for example, have 4.6 million base pair length DNA. 4.6 million, that is 4 into 6, 4.6 into 10 raised to 6 base pair. Haemophilus influenzae, it has 1.8 million base pair DNA. A typical bacterial chromosome contains few thousand different genes. Structural gene sequences, that is the genes which encode proteins, account for the majority of bacterial DNA. Non-transcribed DNA between adjacent genes are termed as intergenic sequences or intergenic regions or spacer elements or spacer DNA. This is non-transcribed DNA present between two genes, or which separate two genes from one another. The sequence is known as intergenic regions. This is bacterial chromosome. This is origin of replication, that is the site where replication starts. These are the key features of the bacterial genome. See, now this sign indicates here repetitive sequences are present, and this single lines indicates that the genes are present, and in between are the intergenic sequences. Now the most but not all bacterial species contain circular DNA. Typical chromosome is few million base pair in length. Most bacterial species contain single chromosome. Several thousand different genes are interspersed, that is distributed throughout the chromosome. Several genes are present onto the bacterial chromosome and distributed throughout the chromosome. One origin of replication is required to initiate DNA replication. Thus, origin of replication is few hundred nucleotides long, Short repetitive sequences may be interspersed throughout the chromosome. These play a role in DNA folding, DNA replication, and gene expression. Remember, repetitive sequences, they are required for folding of the DNA, that is packaging, replication, and gene expression. To fit within the bacterial cell, chromosomal DNA must be compacted about 1000 folds. This involves formation of loops. So this is circular chromosome. Imagine circular rubber band. We form a loops of this rubber band and secure these loops at the center with the thermocol balls. This will look like this looped chromosomal DNA with the associated proteins. Now these proteins are basic proteins. Basic, that is, they are rich in positively charged amino acid. So they bind to the negatively charged DNA firmly and helps in compaction of the DNA. Now see the size difference of the DNA. This circular chromosomal DNA size is almost 50% reduced by forming loops of the DNA. The number of loops varies according to the size of bacterial chromosome and the species. E. coli has 5200 loops with the 40,000 to 80,000 base pair DNA in each. DNA supercoiling is second important way to compact the bacterial genome. Now this looped chromosome, each loop is again coiled many times or twisted in circular fashion and forms supercoiled DNA like this. Again this reduces the size of the DNA. Supercoiling within loops creates more compact DNA. Supercoiling is controlled by two enzymes. First is DNA gyrase, which introduce negative supercoils using energy from ATP, and this also relax positive supercoils when they occur. Then topoisomerase 1 relaxes negative supercoils. See, these two enzymes, they work in opposite manner. DNA gyrase or topoisomerase 2 introduce negative supercoils and topoisomerase 1 relax negative supercoils. In next lecture we'll see eukaryotic chromosome.